In previous videos, we've covered the twin-tail morphotype extensively, and in this video, we'd like to touch on the dorsal-finless phenotype. Strains such as ranchu lack dorsal fins and exhibit bifurcated caudal fin. Such an exceptionally rare combination is nearly non-existent, with equivalent mutations in other vertebrate lineages being extremely rare. This rarity makes goldfish a particularly intriguing subject for the historical and modern research. Historical record and phylogenetic analysis suggest that the twin-tail and the dorsal-finless trait emerged quasi-simultaneously during the domestication process. This process, spanning less than 600 years, led to the genetic fixation of these traits in strains like the dorsal-finless goldfish. At the genetic level, we have identified the coding locus as the key gene responsible for the twin-tail phenotype. The coding gene plays a significant role in dorsal ventral patterning. The stop codon containing allele, the coding mutant, was found to be the responsible allele. Notably, this genotype homozygous of the coding mutant is shared across all investigated twin-tail goldfish strains, including Ryukin, Oranda, and Ranchu. Genome-wide association studies have been employed to identify candidate loci potentially linked to the dorsal-finless phenotype. In simpler terms, the genetic backgrounds of various goldfish strains have been examined using multiple approaches. However, despite this progress, a crucial question remains. How did breeders achieve such rapid genetic fixation of these complex mutations? In this video, we provide a detailed explanation of the genetic underlying the dorsal-finless phenotype with a specific focus on the coding locus. Since all modern dorsal-finless goldfish strains share both the twin-tail phenotype and the homozygous mutation of the coding mutant allele, we hypothesized that the coding locus might be associated not only with the bifurcated caudal fin but also with the dorsal-finless trait. To test this hypothesis, we conducted genotyping and phenotyping of backcrossed progenies derived from hybrid between the single-tail wild-type common goldfish and the ranchu strain. As you can see here, this experiment setup allows us to examine the genetic inheritance pattern of these traits. Let's explain this genetic experiment in a bit more detail. We conducted a backcross analysis using F0 parent from wild-type and ranchu strains. As shown here, the F0 parent had distinct coding genotypes. This initial pairing yielded 123 F1 progenies across 4 clutches. These progenies carried a heterozygous genotype. This means these progenies have wild-type and mutant coding alleles. And these progenies exhibited single median fins including dorsal, anal, and caudal fins closely resembling the wild-type goldfish phenotype. From this result, we concluded that the wild-type coding allele effectively suppressed the dorsal-finless phenotype in F1 progenies. Next, the F1 progenies were backcrossed with the ranchu strain to generate backcross progenies. 2 clutches of backcross progenies were obtained, providing phenotype and genotype data from 116 individuals. As you can see here, the backcross progenies exhibited significant variation in their caudal and dorsal fins. In terms of caudal fins, some individuals displayed single fins while others exhibited bifurcated caudal fins consistent with previous findings. Variations were also observed in the dorsal fin phenotypes. While many backcross progenies lacked the dorsal fin rays entirely, similar to the ranchu strains, as exhibited wild-type-like dorsal fin. Interestingly, we also observed intermediate phenotypes such as an individual with rudimental dorsal fins or reduced number of dorsal fin rays. These findings highlight the complex genetic interactions that govern these phenotypes, offering deeper understanding of how genetic variations contribute to the development of highly specialized traits. To further understand the influence of coding wild type and coding mutant alleles on the dorsal fin phenotype, we analyzed the backcross population. Specifically, we counted the number of dorsal fin rays and genotypes the coding locus in 116 progenies. Our analysis revealed that the coding wild type allele significantly impact the dorsal-finless phenotype. When examining dorsal fin rays, coding mutant homozygous genotype individuals fell into two categories: those with wild type dorsal fin and those with mutant dorsal fins. Wild type goldfish typically have 17 to 22 dorsal fin rays during the late larval stage. Using this as a criterion, we classified individuals with fewer than 17 dorsal fin rays as having a mutant phenotype. Our findings showed that more than half of the coding mutant homozygous genotype progeny exhibited mutant dorsal fin phenotypes. In contrast, coding heterozygous individuals tend to show wild-type dorsal fin morphology, though some exhibit uncategorized mutated phenotypes. These results strongly suggest that the coding wild-type allele suppress the dorsal-finless phenotype, highlighting the genetic link between the coding locus and the mutation in median fins. Since correlation has been identified, we now aim to further investigate whether the wild-type allele of coding truly has an effect. We designed a rescue experiment using messenger RNA microinjection in ranchu progenies. This approach was previously shown in a rescue twin-tail phenotype. In this experiment, we injected coding wild-type messenger RNA into fertilized eggs from ranchu parent as illustrated here. Our rescue experiment provides compelling evidence that the coding wild-type messenger RNA microinjection altered the phenotype of early-stage larvae of ranchu compared to control groups which included non-injected or water-injected ranchu larvae. Specifically, we observed differences in the median fin fold morphology at the pre- and post-cloacal level as shown here. Control larvae consistently exhibit a ventralized phenotype. This included bifurcated or disrupted fin fold on the ventral side. In contrast, messenger RNA-injected larvae display a range of phenotypes. Importantly, some larvae showed significant rescue of the median fin fold phenotype on both the dorsal and ventral sides. Now that we have determined that our messenger RNA microinjection experiment affects the larvae, let's take a closer look at this in more detail. We categorized both control and messenger RNA injected larvae into three groups based on their dorsal fin fold morphology. Group 1, no dorsal fin fold at the yolk and post-yolk levels. Group 2, partial dorsal fin fold at the yolk at post-yolk levels. Group 3, wild-type equivalent dorsal fin fold at the yolk and post-yolk levels. We raised these categorized fish individuals. At the late larval stage, these early categorization aligned with the development of dorsal fin rays. So, what do the results tell us? Group 1 individuals lacked dorsal fin rays entirely. Group 2 individuals exhibited missing rays at several body levels, and Group 3 individuals developed complete dorsal fin rays. This concordance emphasizes a critical role of coding wild-type messenger RNA during embryonic development in shaping the skeletal morphology of the dorsal fin. To further analyze these findings, we compared the messenger RNA injected larvae within each clutch. The result revealed a significant reduction in group 1 individuals among messenger RNA-injected embryos. While the degree of response varied slightly between clutches, the outcomes were consistent across all 4 independent experiments. Taken together, the result from the messenger RNA microinjection rescue experiments and the backcross segregant analysis strongly indicate that the presence or absence of the coding wild-type allele profoundly influence the dorsal-finless phenotype. Bioexperiments, we demonstrated that the coding gene is responsible for both the twin-tail and the dorsal-finless phenotypes. This suggests that the coding has pleiotropic functions influencing the development of all median fins. Here the term pleiotropic function or pleiotropy refers to the single gene affecting multiple phenotypes. Based on our findings, we propose a hypothetical evolutionary process for the dorsal-finless phenotype in ornamental goldfish. Before the twin-tail phenotype became genetically fixed, the penetrance and expressivity of the dorsal-finless phenotype were suppressed by the presence of coding wild-type allele. In other words, even if a genetic mutation causing the loss of the dorsal fin existed, such a phenotypic mutation could not appear due to the stable dorsal ventral axis formation mechanism established by the coding and its related genes. After the genetic fixation of the twin-tail phenotype, the absence of wild-type coding allele allowed for increased penetrance and expressivity of dorsal-finless phenotype. In certain goldfish lineages, breeders and fanciers selectively favored individuals expressing the dorsal-finless phenotype. And eventually, the Ranchu group characterized by both the twin-tail and the dorsal-finless phenotypes becomes prominent enough to depicted in early paintings. Let me address a critical point here. In our study, we have shown that the wild-type allele of coding suppressed the genetic mutation responsible for the loss of the dorsal fin. However, we have not yet succeeded in identifying other genetic mutations that cause the loss of the dorsal fin. Interestingly, 2 independent groups have published studies on this topic. One group proposed that the LRP6 gene plays a significant role, while the other pointed to the DHFR gene as being important. Therefore, there remains considerable uncertainty regarding the genetic basis of the dorsal-finless phenotype and the involvement of these genes. This will likely become an important focus for future research. There are still many mysteries surrounding the dorsal-finless phenotype. However, considering the dorsal-finless phenotype does not appear without the twin-tail trait, it is plausible that the genetic variations related to the dorsal-finless phenotype were accumulating without affecting the dorsal fin. Once the twin tail became genetically fixed, the dorsal-finless phenotype suddenly emerged. This hypothesis could explain why lineages such as Ranchu were established in such a short period of time as we proposed. The relationship between caudal and dorsal fin mutation may not be immediately apparent when observing the adult morphology of goldfish, but it becomes much clearer when examining the embryonic development process. Let's take a closer look at this once again. The embryonic proximity of dorsal and caudal fin may explain their simultaneous mutations. Studies in zebrafish show that while mesenchymal cells of these fins originated from different somite levels, their epithelial cells are connected during embryonic development. This means that although the dorsal fin and caudal fins appeared to be completely separate structure in adult goldfish, examining their embryonic development reveals that these parts are connected. It is therefore not surprising that mutations in the same gene can influence both fins. If we were trying to understand the variation of goldfish morphology using the same logic as we apply to variation in industrial product, we might mistakenly assume that each part of the body corresponds to a specific gene. However, in living organisms, many cells share the same genetic set, and these cells undergo developmental processes to grow and form different shapes. As a result, a single gene can influence multiple parts of the body, demonstrating a phenomenon of pleiotropy.